Put all your beliefs into harmony with science. There can be no opposition, for truth is one. When religion, shorn of its superstitions, traditions, and unintelligent dogmas, shows its conformity with science, then will there be a great unifying, cleansing force in the world, which will sweep before it all wars, disagreements, discords, and struggles. And then will mankind be united in the power of the love of God. Abdul Baha.
Thank you. So today we're so happy to have Dr. Jena Khadem, and she'll be talking about the harmony of science and religion, ethical challenge of biotechnology and genetic engineering. Dr. Khodadad is a fourth-generation Iranian Baha'i. The major part of her life was in the United States, where she received her Ph.D. from Northwestern University in biological sciences, with a focus on cellular and molecular biology, followed by postdoctoral fellowships and faculty positions in medical and graduate schools. Her academic career entailed lecturing to medical and graduate students and research in cellular and molecular biology. Jena is an emeritus professor. Her scientific publications are in scientific journals and academic books. Aside from her scientific research and publications, she's deeply interested in studies centered on Baha'i history, worldview, and the fundamental principles of the Baha'i Faith, such as the harmony of science and religion. In this respect, her latest publication is a book entitled Scientific Principles at Work in the Worldwide Advancement of the Baha'i Faith. So with that, I'll hand it off to Dr. Khodadad.
Thank you very much for this introduction. And I've been looking forward to this presentation because it's in my heart. This theme has for long engaged me. So I'll start this presentation, a PowerPoint presentation, by sharing my screen, and I hope it goes as smoothly as I anticipate. Reflections on the Baha'i principle of the harmony of science and religion. This presentation is inspired by one of the fundamental principles of the Baha'i Faith, the harmony of science and religion. At this beginning, I wish to offer a few introductory words on the Baha'i Faith, the most recent of the world's religions. The principles and teachings of the Baha'i Faith inspire and guide this global age. The Baha'i Faith is widely spread across the entire planet, from pole to pole and in all corners of the world, including within its fold members from diverse races, religions, nationalities, and ethnicities. It is a veritable microcosm of humanity, dedicated to the principle of the oneness of humankind. Thus, it is well suited for bringing about the unification and solidarity of the races, ethnicities, religions, and nations across this planet. This talk is centered on the Baha'i principle of the harmony between science and religion. Among other fundamental principles of the Baha'i Faith are independent investigation of truth, elimination of all prejudices, equality of women and men, the oneness of the human race, the recognition of the validity of the major religions of the world, and the dynamic and progressive interrelationship among them. For the first time in the history of religions, the Baha'i Faith, a world religion, pronounces unequivocally the principle of the harmony between science and religion. This fundamental principle is at the center, at the core, at the very heart of this presentation. The harmony of science and religion is an intriguing, a fascinating principle, which for long has been sidestepped and evaded. Now is the time to reflect on this principle. Among the world religions, the Baha'i Faith unequivocally promotes and champions emphatically the harmony of science and religion by designating it as one of its fundamental principles. Thus, in this presentation, we reflect on whether science and religion can continue to work together in harmony in view of the ethical challenge posed by biotechnology, in particular by the biotechnology of genetic engineering, to the principle of the harmony of science and religion. This presentation will employ throughout the use of PowerPoint. PowerPoint facilitates clarity and order. Foremost, it provides a valuable means for inclusion of the sources of information and citations. This talk represents my perspective, my reflections on the Baha'i principle of the harmony of science and religion, a theme relevant to our specific times. At the start, before diving into this theme, few words of explanation are in order as to why I wish to highlight the ethical challenge posed by biotechnology. In particular, this theme is at the center of an important public discourse of our times. The supreme governing body of the Baha'i Faith has encouraged the engagement of Baha'is in public discourse. This talk is centered on one of the outstanding discourses of our times, which relates directly to the fundamental Baha'i principle, harmony of science and religion, hence its emphasis through this presentation. Additionally, the keywords biotechnology and genetic engineering are among the most highly searched keywords in YouTube searches. Thereby, I wish to offer my understanding, my perspective on this highly searched, vital, and timely theme. May this presentation generate such interest as to inspire the viewer to explore, investigate, and reflect on the Baha'i Faith and its fundamental teachings. The principles of the Baha'i Faith are precisely in tune with this global age. First, I wish to reflect on the Baha'i principle of the harmony of science and religion. Next, I plan to bring to the attention of the viewers the ethical challenge posed by biotechnology, notably by biotechnology of genetic engineering. In conclusion, I wish to call forth reflection on the stance of the Baha'i Faith on the harmony of science and religion. The ethical challenge posed by biotechnology compels our in-depth reflection. On the ethical challenge posed by biotechnology, notably by the biotechnology of genetic engineering. Such reflections lead to a fundamental question. Are science and religion in opposition to one another, or are they in harmony with one another? Science and religion each has been a significant force in human history, essential to the development of societies, civilizations, and cultures. Yet often, science and religion are viewed in opposition to one another. Science and religion are as two mighty forces, as two waves surging toward the development and advancement of civilizations. Thus, harmony of science and religion inspires an in-depth reflection. Science and religion are as seemingly divergent waves that originate from the same source. They arise from the same source as two mighty waves. These two waves, religion and science, cannot be separated. They are intertwined with each other. They are as two waves, two mighty forces in the development and progress of civilizations. Two sweeping divergent waves arising from the same source. The sweeping waves of science and religion originate from the same source. Religion and science are not separate from each other, rather they are intertwined. Indeed, they may be viewed as mighty waves that move onward. As convergent waves, they progress toward the advancement of civilization. Should these sweeping waves converge in harmony, they can generate a tremendous force, lending a powerful impetus to the advance of civilizations. Historically, science and religion have been viewed in opposition to one another. I wish to emphasize that the fundamental principle of the Baha'i Faith, the harmony of science and religion, is emphasized and affirmed by the following few selections from the Baha'i sacred writings. Religion and science are intertwined with each other and cannot be separated. These are the two wings with which humanity must fly. Religion must be reasonable. It must agree with science perfectly, so that science shall sanction religion and religion sanction science. If we say religion is opposed to science, we lack knowledge of either true religion or true science, for both are founded upon the premises and conclusions of reason, and both must bear its test. Enlightened and inspired by these writings, we can pose the key question: are science and religion dissonant or in harmony? Science and religion, dissonant or in harmony, is a question that over centuries has been the cause of much contention, a contention that has continued to the present, a time when great advancements in science, technology, and innovations have been achieved. A related question is, are science and religion at odds with one another, or are they complementary and supportive of one another? Both science and religion are centered on what is believed to be the truth, in one respect relating to physical phenomena, and in other respect relating to spiritual truths. Thus we ask, are science and religion in competition, or in cooperation, or in synergy? Are they complementary and supportive of one another? We can probe the question of the harmony between science and religion through several approaches. Traditionally, for long, it was assumed that science and religion are at opposing poles, and that never shall the twain meet. Science relates to the operation of those universal laws of nature. For those who believe in a religion, religion relates to spiritual laws and principles. One of the fundamental reasons that religion and science have been viewed in opposition to one another is due to lack of understanding or misinterpretation of metaphors, analogies, and similes used in religious texts. These have been taken literally, though they hold profound insights and have layers of meaning, whose depths may be fathomed through thoughtful reflection. Lack of understanding of numerous metaphors in the Judeo-Christian as well as in other sacred texts, Zoroastrian, Hindu, and Muslim, are factors that have contributed to such misinterpretations. Historically, on one hand, contention between science and religion has deterred women and men of science from partaking of the wisdom and riches that religions can impart. On the other hand, it has hindered many women and men dedicated to a religion from the enrichment that science can impart toward understanding the mysteries of life and enhancement of the quality of life. At one time, a talk on science and religion would have likely centered on a literal understanding of certain religious texts, such as the story of creation in 7 days, the Garden of Eden, the question of evil, and numerous other examples. Abdul Baha, one of the central figures of the Baha'i Faith, through his talks and writings, sheds insights on these things. Some of these are to be found in a remarkable book known as Some Answered Questions. He sheds insights by explaining the inner meaning of these metaphors. Yet there are those who insist on the inerrancy of a literal understanding of the sacred text, such as the story of creation, exclusivity of salvation, and others. In our times, the literal understanding of such metaphors no longer merit serious consideration. Interpretations of sacred texts go well beyond the literal. And in current times, we are well beyond a literal understanding of such metaphors and analogies. There are novel challenges to be met in our times. In particular, advancements in science and technology compel insightful reflection on the Baha'i principle of the harmony of science and religion. On the Baha'i principle of the harmony of science and religion. During our times, special challenges are posed to this principle, in particular by ethical challenges posed by biotechnology. Among these biotechnology, notably by the biotechnology of genetic engineering. Now, this prompts an in-depth reflection on the biotechnology of genetic engineering that is regarding ethical and spiritual standpoint in bringing together material and spiritual civilizations. I quote from Baha'i sacred writings. Although material civilization is one of the means for the progress of the world of mankind, yet until it becomes combined with divine civilization, the desired result, which is the felicity of mankind, will not be attained. In our times, momentous advancements have been achieved in biotechnology. This prompts us to ponder whether biotechnologies hold prospects for human well-being. Their benefits are significant, among them identification of the genetic base of disease, eradication of certain genetic diseases, development of therapies, prolongation of human life, and much more. Or whether biotechnologies foreshadow unfavorable outcomes toward human detriment. Few among their unfavorable, even ominous implications are human cloning, eugenics, and generation of biohazards. As the gene is at the foundation of heredity, we are faced with multitude of issues and concerns. At this point, it is important first to briefly overview some of the historical background on human heredity. Early groundbreaking studies include the work of Charles Darwin, with its implication on inherited traits presented in 1856, the work of Gregor Mendel on pea hybrids presented in 1863. William Bateson foresaw that the science of heredity would lead to great discoveries and that its power would be applied to control the composition of a nation. Thus, Bateson introduced the concept of eugenics. Whether such control would ultimately be good or bad for that nation or for humanity at large was considered a separate question. Francis Galton coined the term eugenics in 1904. Eugenics may be defined as the science of improving a human population by controlled breeding to increase the occurrence of desirable heritable characteristics. Eugenics has horrific implications. A few years later, in 1912, a German physician, biologist, eugenicist, came with the term racial hygiene. And that was Alfred Plotz, and promoted the horrific concept of racial cleansing in Germany. In fact, the United States has also been tainted in respect to eugenics. Genetic cleansing was promoted between 1927 and 1972 through the sterilization of some women identified as imbeciles. Regarding this case, known as Buck versus Bell, 1922, the US Supreme Court ruled that Virginia law was constitutional and that Carrie Buck should be sterilized. Her sterilization was the first of approximately 8,300 performed under the law between 1927 and 1972. Eugenics has horrific implications. At this point, I wish to take a brief look at certain landmarks in the development of the science of heredity. Early on, Aristotle recognized that heredity is as a current of information moving across generations. Gregor Mendel, in 1866, introduced Mendelian genetics with his work on pea plants. However, the structure of DNA in 1953 was revolutionary. It led to molecular genetics. Molecular genetics has ever since dominated the field of genetics with its revolutionizing effect, leading to the development of some remarkable biotechnologies. With the emergence of molecular genetics, we are faced with a challenge. Challenges that on one hand are auspicious with immense possibility, yet on the other hand, they pose the dilemma. Thus, they pose both the challenge and the dilemma. The new discoveries in biotechnology were hailed with great enthusiasm by some passionately as the ultimate experiment, man-made revolution, man-made evolution, the brave new world of genetic engineering. The Brave New World refers to a dystopian novel written by the English author Aldous Huxley in 1931 and published in 1932. This book was published some 20 years before the structure of DNA was deciphered. Aldous Huxley foresaw that which was on the horizon could lead to a dystopian future with its horrific implications. Yet others foresaw the new findings auspicious, a boon to humankind. In time, it led to the discovery of the structure of DNA in 1953. That discovery was enthusiastically hailed as alchemy revived, improving nature, harnessing the gene, and remaking the world. Why such discordant views? Which of the two did the new discoveries portend? A blessing, boon, or the dismal gloom? Which is it? Could it be both, boon and also gloom? As it relates to the theme of this presentation, the harmony of science and religion, boon and also gloom? First, I plan to briefly overview the boon, the advantages, the favorable outcome of this biotechnology, the benefits, life-saving benefits of some of the discoveries, advances at the forefront that have been made possible through biotechnology. Next, I plan to reflect on the possible disadvantages, the unfavorable outcome of biotechnology, that is the gloom, harmful effects which can last over generations, extending beyond this to future generations. Now, an overview of certain advances at the forefront in biotechnology. Some of these advances are a boon, yet they hold outcomes that are beneficial to human well-being. Some are dismal gloom, they portend detrimental outcomes. Now, an overview of advances at the forefront. The discovery of the structure of DNA in 1953. Several biotechnologies, among them recombinant DNA, gene cloning, stem cell research, and genetic engineering, hold prospects for human well-being, yet can also have detrimental outcomes. Their significant benefits include identification of the genetic diseases, development of therapies, prolongation of human life, and much more. Among their unfavorable implications are eugenics, gene enhancement, human cloning, and the generation of biohazards. These advances, with their significant as well as their unfavorable implications, are the outcomes of the discovery of the structure of DNA in 1953. The discovery of the structure of DNA was revolutionary. In 1962, Nobel Prize in Physiology and Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins, but left out a principal figure, Dr. Rosalind Franklin, who had died in 1958 at the age of 37. This is an exciting, a fascinating story, yet it's egregious. It is fascinating because of how it happened, that aha moment, that eureka moment of sudden discovery. It is egregious because the process ultimately left out the major contributor, Dr. Rosalind Franklin, as one of the recipients of the Nobel Prize. Dr. Franklin had died before the prize was awarded. The Nobel Prize is not awarded posthumously. I wish to relate the fascinating story of this monumental discovery, leading to the eureka moment of that sudden discovery. How so? Researchers in this field, James Watson, and Francis Crick were conducted by Maurice Wilkin, the director of King's College Lab in London, and shown the data studies of Dr. Rosalind Franklin. Let us now look at that key study, the fundamental data, the key image which divulged the structure of DNA. The discovery of the structure of DNA was based on the data, the research of Rosalind Franklin, the X-ray diffraction image of DNA. Viewing this image led to that eureka moment for Watson and Crick. Over the years, various models of DNA had been proposed as a single strand or as a triple strand, and so on. However, it was the data, the crystallographic image of DNA of Rosalind Franklin, which at long last divulged the model of DNA. It became suddenly clear that DNA must be in form of two strands, the double helix. How so? This was based on the observation of this image, the image of DNA through X-ray diffraction studies of Rosalind Franklin. The distinctive X in this image divulged the pattern, a double helix. So James Watson and Francis Crick became excited and giddy after viewing this image of DNA of Rosalind Franklin. They now had the view of the essential image of the molecular structure of DNA. They had that eureka moment. Immediately, they perceived the overall organization of the structure of DNA. Over time, eureka moments have sparked scientific discoveries. Some of us may have experienced a eureka moment when suddenly we become aware of a long search for reality, whether a scientific discovery or a spiritual discovery. Through model building, things fell into place. The double helix structure of DNA of two strands was at long last described. So what did James Watson and Crick do? Through model building using hammers and screws and metal plates, they unraveled the organization of the double-stranded model of DNA. A photograph of that model building is in view. It shows Watson and Francis Crick in state of awe and euphoria. All that model building. Thus, James Watson and Francis Crick are credited with deciphering the structure of DNA with that big eureka. Then James Watson and Francis Crick went to the local pub, Eagle Pub in Cambridge, near Cavendish Laboratory, to celebrate. A sign has been put up by Eagle Pub in Cambridge acknowledging and celebrating that historic event. The double helix at the local pub in Cavendish. This pub to this day bears the distinction of its association with the discovery of the structure of DNA. They use the wording for DNA double helix, the secret of life. That was where they first announced. So that was what happened. That was what happened on February 28, 1953. That Francis Crick and James Watson first announced their discovery of how DNA carries genetic information. The DNA double helix holding the secret of life. Having related this story, in the interest, I'm not sure in this setting, the rather complex yet magnificent molecular structure of DNA. Therefore, in the interest of avoiding undue complexities in this session, I will bypass showing and explaining the diagram of the molecular structure of DNA. For now, the discovery of the structure of DNA, the secret of life, had a great impact, a revolutionary impact. Once the molecular structure of DNA was deciphered, it led to development of biotechnologies with their immense possibilities. Biotechnology holds possibilities to improve human life and also, in some respects, to deteriorate the condition of the human species. Biotechnologies developed in the 20th century hold possibilities then to improve human life or deteriorate the conditions of the human species. The discovery of the structure of DNA in 1953 spurred a revolutionary progress in biotechnology. Biotechnology holds prospect then for the welfare of humankind, but also portends detrimental outcomes. Their benefits are significant. Among their benefits are identification of genetic base of disease, eradication of genetic diseases, development of therapies, prolongation of human life, and much more. Few among their unfavorable implications are human cloning, eugenics, and biohazards. Among biotechnologies developed in the 20th century are several to be noted. They include recombinant DNA, gene editing, stem cell research, and reproductive cloning. Some of these technologies hold prospect for the well-being of the human race, but can also have detrimental outcomes. Among their significant benefits of the genetic base of disease, eradication of certain deadly diseases, development of drugs and therapies, prolongation of human life, refinement of human genome, and much more. At this point, I wish to offer a few words on the intricate and elegant recombinant DNA biotechnology. This biotechnology has been a tremendous boon to humankind. Thus, it warrants brief explanation. This brilliant, intricate, and significant technology has made possible effective novel treatment for a number of diseases. Recombinant DNA technology. Recombinant DNA technology is an elegant biotechnology which has been a tremendous boon to humankind. Thus, it warrants a brief explanation. This brilliant, intricate, and significant technology has made possible effective treatment for a number of diseases. It has been a boon to humankind. Briefly, two sets, that is, different segments of DNA, one from the host and the other the desired DNA, are combined under specific conditions. And this is a brief coarse diagram of the recombined structure. The diagram shows the two segments of DNA in two colors. This is known as a chimera because it is the result of two genetically distinct sources. Recombinants are treated in such a way to significantly increase their number. By this means, through this technology, a large amount of the desired product coded by the inserted DNA can be obtained in pure form. This technology has been life-saving. Through this technology, vital therapeutic agents can be obtained in large amounts in pure form, such as blood clotting factor 8, deficient in hemophilia, insulin, and an ever-growing list of therapeutic agents. The importance of this significant biotechnology cannot be overemphasized. This fascinating technology is revolutionary. It provides vital agents for the treatment of a number of diseases. Thus, we can conclude that recombinant DNA technology is beneficial, therapeutic, life-saving, and highly moral. The early recombinant DNA experiment. At this point, I wish to introduce the humor, the significance, and the ethical concern which have been historically associated with this type of experiment. The early recombinant DNA experiments were historic and noteworthy in several respects. In 1972, Paul Berg created the first recombinant DNA chimera at Stanford. It was the result of combining DNA from two genetically distinct sources. 2 years later, in 1974, Cohen and Boyer created an amusing chimera. They transferred frog genes into bacterial cells. It came to be known as the Frog Prince experiment. Aside from the strange humor therein, much learning has been gained from this experiment. Furthermore, this type of experiment also raised alerts about several ethical issues. Importantly, from the recombinant DNA experiments, much has been gained, specifically toward the treatment of a number of diseases and human well-being. Aside from the seeming absurdity of mixing the DNA from two species and the humor therein, the development of this type of technology, that is recombinant DNA technology, has been a boon. This technology serves for the synthesis and production of a number of vital therapeutic agents in highly purified form. Indeed, we can build a strong case that such technologies are a boon to humankind. Such technologies serve for the betterment of humankind. Thus, they can be highly moral and ethical. Thus, through recombinant DNA research, much has been achieved. As an example, in order to obtain insulin essential in diabetes, one route was to obtain pancreas from large number of cows. Pancreas is where insulin is produced. It was necessary to grind the collected pancreas organs, isolate and purify the insulin fraction. However, by employing recombinant DNA technology, large amounts of insulin can be obtained in highly pure form. Nevertheless, we can rightly ask, can such technologies lead to an ethical outcome? The first recombinant DNA experiment raised alerts about the ethical issue entailed in such experiment. It is heartening that the scientists themselves were at the forefront. Hoiberg, a leading researcher in the field of recombinant DNA and one of the recipients of the 1980 Nobel Prize in Chemistry, creator of the first DNA chimera, first expressed concern. He was alarmed by the inherent hazards of recombinant DNA technology. Together with several other noted scientists in the field, he raised serious concern. This led to 2 conferences in Asilomar. To the second conference in 1975, lawyers and journalists were also invited. There was transparency. Berg summarized the data and outlined the scope of the problem. It was a volatile meeting. Lawyers spoke, scientists spoke, a unanimous decision was reached to place a temporary moratorium on this type of research, on this recombinant DNA research. Other powerful genetic technologies were developed in the 21st century. These technologies have spurred scientific and medical revolution. One was the decoding of the entire human genome in 2003. At the beginning of the 21st century, a bare 50 years since the discovery of the structure of the DNA molecule. This was the Human Genome Project, an international scientific research endeavor. It was formally launched in 1990, cost $3 billion, and was declared complete in 2003. We now know that the human genome includes over 3 billion letters of DNA. It consists of 23 pairs of chromosomes and codes for 20,687 genes in total. It accommodates enough variation to make each of us distinct and yet enough consistency to make each member of our species different from other species. The decoding of the entire human genome has been significant and extremely beneficial to human well-being. Once the human genome was deciphered, several significant technologies developed, notably among them gene copying. Several significant fundamental technologies led to the technology of genomic surgery and gene editing. Among these, one in recent times that has become quite familiar to the public is known through the acronym CRISPR-Cas9. CRISPR-Cas9 is an elegant technology for gene editing. The work was revolutionary. There have been legal cases on who should receive the patent for it. This powerful technology enables genomic surgery gene editing. A technique for gene editing. The possibility of CRISPR are immense. If we know the DNA sequence related to a given organism, we can perform genomic surgery. The development of this technology for gene editing was the outcome of the research of a number of scientists. Among them, two remarkable women scientists at the forefront made significant contributions. Co-inventors of CRISPR technology. The 2020 Nobel Prize in Chemistry was awarded to two women scientists for their groundbreaking work on gene editing, Jennifer Doudna and Emmanuel Charpentier. Dr. Jennifer Doudna from University of California, Berkeley, and Dr. Emmanuel Charpentier, director of Max Planck Institute. There were other researchers who made significant contributions to this field. The possibilities of CRISPR are enormous. Among these possibilities, some offer remarkable benefits with significant implications in the treatment of diseases and abnormalities. Some have worrisome implications as to their misuse. This technology enables genomic surgery. With genomic surgery, it's possible to chop, delete, add to, and revise the gene at will. This technology has the potential to eliminate certain genetic diseases. It enables development of novel therapies. However, this technology has also worrisome implications. Among them, eugenics, already explained, creation of biohazards, and their release into the environment. These worrisome implications also include generation of gene drives. On one hand, these biotechnologies cannot be overlooked nor eliminated, as they can offer numerous benefits to humankind. Yet, on the other hand, ethical and spiritual concerns continue to engage biologists, ethicists, and others. Concerns ethical and spiritual. Significant ethical and spiritual concerns continue to face us with ethical and spiritual implications. Among these concerns are eugenics, already explained. Gene enhancement enables the selective enhancement of a special trait or characteristic. Thus, it can result in inequalities, evolution of a super race and underclass. This brings to mind the emerging realities predicted by Aldous Huxley in his dystopian book, The Brave New World. Some associate this technology with creating what has been called designer babies. And reproductive cloning by two means, using two possible approaches through use of somatic cells, cell other than the germline, or by the use of embryonic stem cells. Both of these approaches, reproductive cloning biotechnologies, provide benefits, but also have ethical implications. In view of the lure and fascination of the most recent powerful genetic technologies developed in the 21st centuries, we can rightly ask whether scientists can or will continue to self-regulate. The ethical and spiritual implications of these technologies compel this question: can scientists self-regulate? To what extent should spirituality, moral principles, and ethics come into play? More importantly, how can they be enforced? How can they be regulated? Several fundamental ethical concerns emerge. Indeed, at this point in time, we are at the crossroads of science, ethics, and spirituality. At the crossroads of science, ethics, and spirituality. This compels us to reflect on the standpoint of the Baha'i Faith in respect to the harmony of science and religion. This compels us to ask the question: are we moving closer to the brave new world of Aldous Huxley. Earlier in this talk, I made reference to the dystopian book of Aldous Huxley, The Brave New World, wherein Huxley anticipated ominous circumstances and scenarios. Can these indeed become realities? Or we ask, are we advancing toward the vision foreshadowed by Shoghi Effendi, the appointed guardian of the Baha'i Faith? Are we advancing toward the vision foreshadowed by Shoghi Effendi? We have the guidance through the writings of Shoghi Effendi in the World Order of Baha'u'llah regarding a world system guided and inspired by the vision expressed through the following guiding statements in the World Order of Baha'u'llah. The enormous energy dissipated and wasted on war, whether economic or political, will be consecrated to such ends as will extend to the range of human inventions and technical developments, to the increase of the productivity of mankind, to the extermination of disease, to the extension of scientific research, to the raising of the standard of physical health, to the sharpening and refinement of the human brain, to the exploitation of the unused and unsuspected resources of the planet, to the prolongation of human life, to the furtherance of any other agency that can stimulate the intellectual, the moral, and spiritual life of the entire human race. This is the vision. Fundamentally, such reflections lead to the reality of our times, that there is significant imbalance and discordance between advancements in science and advancement in spirituality. Spirituality has not advanced in line with advancement in science and technology. These have not been in synchrony. Science and spirituality have not advanced, progressed in tandem. We have progressed materially, but not spiritually. Some may even hold that we have advanced in science and technology, but have indeed regressed in spirituality. Material and spiritual advancement must go hand in hand. Both are essential to the progress of civilization. Material civilization is like a lamp, but the spiritual civilization is like the light in the lamp. This lamp without the light is a useless thing. Now, at the conclusion of this presentation, we continue to ponder, to reflect on this Baha'i principle of the harmony of science and religion and the ethical challenge posed by biotechnology. This presentation was centered on reflections on the fundamental Baha'i principle of harmony of science and religion and the ethical challenge posed to it by biotechnology. This presentation was based on some of the current knowledge in the field of biotechnology. With the passage of time, the increasing development of novel biotechnologies are bound to provide yet greater benefits. They're also bound to pose yet new and greater challenges. Once again, the viewer will be left with that fundamental enigmatic question: does biotechnology serve as a boon or gloom? On one hand, novel biotechnologies, through the great benefits they impart, are bound to serve as boon to humankind. Yet on the other hand, they may pose more serious concerns, the gloom. Biotechnologies hold enormous value, yet they can also pose serious challenges. May humankind advance in line with that higher level of spirituality, conducive to an ever-continuing boon toward human well-being and felicity. At this, the conclusion of this presentation, I wish to express my appreciation to the viewers of this presentation for their interest and investment of time in listening to and viewing this presentation, which was centered on one of the greatest scientific discoveries and discourses of our times. I appeal to the viewers to reflect on the implication of the fundamental Baha'i principle of the harmony of science and religion for this and future generations toward the progress and well-being of humankind. As biotechnologies continue to advance, new possibilities, new challenges, new questions, both beneficial as well as dire, will continue to engage and intrigue humanity. Will such ever ongoing advances in biotechnologies continue to serve as a greater boon or more dismal gloom? Or will they continue to serve as both boon and also gloom? Such questions underscore the importance of the exercise of caution and also maintaining balance and vigilance toward harmony between advancements in science and advancements in spirituality.
Thank you so much for that really insightful talk. We really appreciated it. Now, if anyone has any questions, you can put it in the chat. First question is, how do we apply the scientific method to religion in an attempt to unify the two ways of thinking about ethical ways of using scientific progress?
Well, the first question, how do we apply scientific method to religion? I think it's already there for people who want to focus on that, because in writings, in scholarship, the scientific method, which is looking at your data and making sure that your data fits your argument, has already been done. It's already in practice. I don't know if I've answered the question of the person who asked the question. Always making sure that we have the correct data, we have the correct information, and we build our argument based on solid foundation. And there are many presenters, many authors of books, many scholars who do exactly that. And the second question was?
How can you unify these two ways of thinking, basically?
How can you unify? Well, the Baha'i Faith has unified that by emphasizing the importance of scientific research and by also introducing the ethics in what we do. The spirituality, the spiritual element, the ethics in our undertaking. This can be unified by individuals who believe in that, who believe in bringing the two together. Now, in our society at large, as I have tried to explain, this is not being done by a number of researchers. And how do you implement it? How do you implement that? That is a really tough question. There are moratoria. Are they going to be followed by researchers throughout the world? That is the puzzle. And by what means can you implement this? By what means can you enforce those? Because right now the technology is such, for example, for CRISPR, it is relatively simple that anyone with a modicum of scientific knowledge in a lab that doesn't need to be terribly well equipped can go ahead and carry on some of these experiments. How do you regulate that? I don't know. I think that is the next challenge at the world level to be addressed. That's my response.
Thank you. The next question is, what are your views regarding the increasing domain of artificial intelligence?
Well, the artificial intelligence is not my area of knowledge. I know just few things about it, possibly not more than those in attendance today. I'm in awe of it, but I'm also in the back of my mind, I'm also concerned by it. But I don't have sufficient information and knowledge about artificial intelligence to give you a meaningful response.
Got it. The next question is there a worldwide accord which dictates how scientific discoveries can be utilized?
There have been attempts, there have been discussions, and they have come up with certain thoughts, certain regulations, but I'm not aware that they're being implemented. Because you're like everything else, I think, as a result of COVID, many of us recognize that it's very difficult to implement certain rules and regulations. And I think similarly in this particular area, it's very difficult to implement that. So this raises the question and the need for world ethics, for a system that could implement and enforce some of these. But at the present time, I'm not aware of that, because any nation at any time can go ahead and do whatever it wills to do.
Okay, well, it looks like that's all of our questions. So thank you so much, Dr. Khodadad, for this great talk. We really appreciate it. Thank you. So now I'll introduce our speaker for next week. Our speaker next week will be Mr. David Langness, and he will be talking about the one unavoidable solution to climate change, a unified world. So again, these talks are every Saturday at noon Eastern time, and I've put the link to our YouTube channel and our contact form if you're not already on it. Thank you so much, everyone, and we will see you next week. Have a good week. Bye.